happy to see you all today. The COBRE on Opioids and Overdose welcomes you today. I'm Tracy Green, and I'm really excited to have a guest lecture here today from Northeastern University, Zhenyu Tian. And we're going to talk about or hear more about non-targeted analysis, and that'll range from environmental to public health related applications in the street drug supply. So really excited to hear from Dr. Tian. And I should mention that he is an environmental chemist and an assistant professor at Northeastern University. And he and his laboratory team focus on the identification and quantification of known and unknown contaminants in our environment to assess their risks to human health and the ecosystem. And so whether it's an environmental pollutant or the street drug supply, you'll hear a lot through this talk, and we look forward to learning from you. Thank you and welcome.
Thank you. Thank you, Tracy, for inviting me and the introduction. So hello, everyone. This is Zhenyu, and today I'm going to talk a little bit about non-target analysis. Some of them, I think the majority would be the environmental application, and small part would be the street drug screening that we did in collaboration with Tracy. Okay, so let me start. Here's the outline. So basically, we will have a very brief introduction and then go to the rubber toxicant study. If you haven't heard about that, that's kind of like a quite interesting story in the environmental side. And then talking about organic contaminants in water, this is really where I was, kind of got interested in the drug of abuse or like drug issue, because we see drug in the water samples all the time. And then finally, that's the most relevant part. Okay, so the reason we do non-target analysis, if you a little bit knowledge about, things like BPA, right? If you have like a bottle, it says like BPA free, right? That's one thing. And then also another bigger thing is PFAS. So these are kind of like the famous contaminants, right? Like some of these, right? Like if you see BPA free, doesn't mean it's bisphenol free, because these days and more they shift to BPS, right? And then PFAS, you can see a lot of applications, not only in our rain jacket or our like pens, but also in makeup, right? So this is something directly in contact with your skin or even mouth. So that's kind of like, the big picture, why we do this, right? Like organic contaminants are everywhere, and some of them are bad with adverse health outcome. So we need this knowledge to identify them, quantify them, and this knowledge will be used for the government for regulation as well as the industry to reformulate, because they also want to make healthier, greener products mostly, right? And then if we go back to why we have to do non-target instead of just looking at the targeted contaminants, it's really because of the increasing use of synthetic chemicals. You can see generally there are tens of thousands of chemicals. Some of them we have a clear category, some of them we don't know. They're just like labeled as industry chemicals, but a lot of them actually goes to our consumer product. So this big number means we cannot analyze them like one by one, because you cannot just like store 10,000 standard in your lab, right? And the other thing is when these chemicals goes into the environment, either they're into the water or soil or our bodies, they don't magically disappear, right? So they will become unpredictable transformation products. Some of them are becoming more toxic, some of them becoming more persistent. So that is why, right, like we are adding some more to this big pool of chemicals. That's why we need a new paradigm. This is called non-target analysis. So the idea is, right, the conventional method is targeted. So it means you have a fishing rod, or you are kind of like fishing the chemicals one by one, or a dozen that you have a established method, you have standard in your lab, you have a calibration curve. But this one is accurate, sensitive, but it's not high, like, the range is not big enough. Instead, the non-target analysis is we don't need to have a standard, we don't need to have a calibration curve, but we can just like try to see as many things as possible. It's almost like using a big net to grab a lot of fish instead of like one by one, right? So this is non-target, and the tool that we use for this is high resolution mass spectrometry. So it can tell you the accurate mass of different molecules. That's why it's possible. And the workflow for non-target looks like this. So if you look into the first two stages, this is quite similar with any type of chemical analysis, right? You go for like sampling, sample pre-treatment, and then instrumental analysis. But what really makes this different is the later stage, right? The data size and the complexity from non-target analysis is really big. So you have to spend a lot of time on these data pre-processing. You want to prioritize your interested features. And finally, when you even have a formula, you have some spectrum, a lot of time these are unknown. So you need to link them with a specific structure. So this is identification. I would say, right, 60, 70% or even more these days are spent in here. And also you need a lot of creativity, how to make this work, how to do the de novo identification. So this is a developing field with a lot of challenges and opportunities. So the first story is going to be, right, like how we use non-target analysis to figure out a acutely toxic chemical in stormwater that kills coho salmon. So this is the work that I did when I was a postdoc in University of Washington. At Pacific Northwest, the salmon, all these species are pretty big thing because they're not only, right, like of fishery value, but they're important part of food chain, right? Like when they are swimming back, they are actually bringing in the nutrient, which is their body, to the land system. So this is quite kind of important. But in Pacific Northwest, especially somewhere near the greater Seattle area, people begin to see, okay, this one species called coho salmon, when they are swimming up the creeks, they die before they can lay their eggs. And this is right, like the time during late October, November. So when it rains, usually this is the water level goes up. This is the good timing for them to swimming up, but they just die in the middle. And the symptom was pretty interesting because it's not like they just like quietly die. They will be struggling very vigorously. So it almost feel like they lost their equilibrium. It's like they're drunk. They will be swimming in circles for a couple of hours until they completely die. So this is attracting a lot of attention because you will see like a dying salmon doing this weird stuff in the creek. So it's been documented since like the 2010s. People are wondering why this is happening, right? And this is a picture I took in 2019 so that you can see, right, like this is a dry period and then a big rain. So a lot of mortality cases, the volunteer called us, we go to the site and we cut open the fish, eggs are in there. So this is pre-spawn, right? This is exactly that problem. And if this trend continues, right, this is the model prediction, the coho salmon in the Seattle area will be die out in just like, 30, 40 years. So this is bad. And people are wondering why. What is causing this problem? And the collaborators before my previous group, these are Washington State University and NOAA, they are biologists and they also use a lot of like modeling, kind of like ecological methods to figure out what is causing this. And they found some of the parameters, especially the urbanization gradient and also like traffic intensity, right? If you have a map based on daily average traffic, and also you have a map of like mortality risk for coho salmon, you will see them like correlating very well. So basically you can use the traffic to predict the mortality risk. And because of this correlation, they're saying, okay, if this is something about the road, if this is something about the stormwater coming off from the road, because of this, they collected, the road runoff from the highways and they exposed to coho salmon, and they found, oh, they did die in the same symptom. So like, okay, this is probably linked to roads and cars, but these are ecologists, these are fish biologists, they're not chemists. The toxicant for them is still unknown. At that moment, they don't even know this is pathogen, biological, or this is chemical. So this is where my previous group, led by Professor Ed Kaloje at UW, we come into this work. So basically, what we do first is use the high resolution mass spec. Remember the non-target analysis, right? We are using this method to analyze stormwater as well as the mortality event related water, right? So if there's a volunteer call us, we go to the site, we pick up the fish, we also collect some water on site. So that we can, right, like analyze all these samples together and see what are the chemicals shared by all this water. So it means whenever there's a fish got killed, right, the stormwater runoff can kill. So it's also here, right? Whenever there's a fish killed, we see this group of chemical. So based on this logic, we are able to narrow down the scope from hundreds of thousands to hundreds or thousands to 57 features. So these are the things we call the cohort mortality signature. And you can see some of these are just pretty generic surfactants. Some of them are like additives for polymers. And then we can actually use this group of features to track the treatment system, right? If stormwater treatment can remove the toxicity, then we should be able to see these things removed. And also, more importantly, we are using this group to track the sources. Where are the toxicity coming from, right? We know it's stormwater, but we want to be more specific. So this is where we use this, right? How we use this signature. All these like lines you can see is one of the fluid that is used for cars, automobile, right? We have all these like oil, antifreeze, and everything, and also we have one that's a tire wear particle. And we did this like kind of unsupervised cluster to see how these things are related to the water sample that kills salmon. And you can see all the other things are not really close. But there's one thing, right? This tire wear particle leachate is grouped together with this like field samples and also pretty close to the runoff. So that get us thinking, okay, chemically, right, these toxic waters are most similar to tire wear particle leachate. We probably should be looking at tires. But at that time, we got some pushback from the industry say, okay, you probably get this some chemicals picked up by the tire from the road. It's not the tire themselves, right? That's a legit argument. So we need to go a little bit further to find out smoking gun. So this is where I spend like more than 2 years to go through the whole process. So the basic idea is if we can start from the tire leachate, and if we can simplify the chemical, which is actually quite complex, right? You can see 2,000, more than 2,000 chemical features in there. If we can simplify this to a few or even one that still reserve the toxicity, then we know that's the causal toxic. There's a smoking gun chemical, right? So basically you can see guided by non-target analysis, we use a lot of chromatography, chemical separation, basically starting from thousands of chemicals to hundreds to just a few. And in this very small part, right, you can see a red magenta color solid that's still kind of like preserve the toxicity. If you dissolve this solid to the fish tank, it kills salmon in just a few hours, faster even than the stormwater. So this is okay, we got it here. But it's still not the end point because even we have a molecular formula, we don't know the structure. We don't know what it is. We don't know where it's coming from. What makes this thing harder is it is a true unknown, which means if you look into the formula for making tire rubber, you don't see the same chemical with this, right, like formula. So you have to do something with different. And my idea was, okay, we actually stuck with this formula for a few weeks. Then suddenly there's a eureka moment. It's like, okay, if we assume this is not a chemical itself, but this is a transformation product, because when the chemicals are used, right, they're going through this chemical reactions. So usually the chemical transformation, the reaction will change the hydrogen and oxygen because these are relatively easier to modify. But the carbon and nitrogen atoms usually are the backbone. They are easy to, they're harder to change. They will preserve. So let's see if we have any chemical with the same carbon and the nitrogen number. Here we go. We find a chemical called 6-PPD. So this is actually a very essential additive for rubber, and not only in tire rubber, because for any of the rubber, right, our sneaker soles, you need to add this chemical to prevent the rubber from cracking, right? This chemical will react with ozone so that the ozone will not crack your long chains of rubber. And you can see, right, they are sold in tons because about 1 to 2% of all the rubber tires are actually made of this chemical. So it's about 1% weight of the tire tread. Okay. So we now have a clue. This is probably something related to 6-PPD. Dig into the older papers. This thing I found, right, this paper from 1983, people are recording some of the ozonation product. One of the things they call this dinitron is actually having the same formula as our unknown. So it's like, okay, this is probably our guy. Let's try to do that. So with our collaborators' help, we are doing ozonation experiment with 6-PPD, and you can see some color change over here. And then you pull this part out, you analyze them with high resolution mass spec. Bingo, you find a match, right? So the same retention time of what we got from tire leachate, also from the 6-PPD ozonation, that's the same thing. And also you will see this mirror plot a lot in the later slides. This is kind of like a early kind of like a knowledge on here, right? So on the top, this is the spectrum from tire leachate. In the bottom, the black one is from the ozonation. And you can see basically they match pretty well. Multiple bars means they are the same thing. So we got it, right? Like this is indeed a chemical from 6-PPD. And later we realized, okay, the 1980s paper was wrong. It's not dinitron, it's actually a 6-PPD quinone. This is another collaborator from University of Toronto. They did a very good NMR analysis, give us the structure like this. So the proposed like pathway is from 6-PPD to 6-PPD quinone. You can see the parent compound is not toxic, right? It's not going to kill fish any fast, but once it's in contact with ozone by a two-step gas phase ozonation, it becomes 6-PPD quinone, super toxic, kill fish in concentration at nanogram per liter. So that's why we see this phenomenon. Okay. So this become a pretty big deal because, right, solve this puzzle, and also it's correlated to, a lot of like Native American like tribes. They are relying on coho salmon, and now they know why their numbers are down. So this is getting some kind of a news, and also potentially causing some formula change in the industry because they realize, okay, this is not good, right? This is a pretty confirmed result. And while this is the previous administration, right, the Congress is also seeking some advice on how this should work. I believe at the state level, especially California and Washington, they are doing something about like the tire formula because they are the most affected states. And so from there, this research is going, out of our control. All these like other countries, right? Like the scientists began to see, oh, if we have a busy highway, we will also detect this thing. And there's more than coho salmon that's sensitive to this chemical. So you can see rainbow trout, brook trout, and some other chars in different continents are sensitive to 6-PPD quinone at sometimes like nanogram per liter, sometimes microgram per liter level. So both like pretty low levels. And also, right, like except for fish, except for salmonids, some of the research found that the degradation product from 6-PPD and IPPD, another antioxidant, could produce some of the endocrine disrupting chemicals. So this is not only to fish, but also potentially to human, potentially to mammals. So these are kind of like ongoing. And some of my recent work, right, because I'm on the East Coast, right, East Coast generally don't have the Pacific salmon species. So we are really not like seeing the mortality cases. But one of the application is because if you think about the artificial turf field, a lot of them having this black kind of like chrome rubber particles in them. These surprisingly are coming from recycled tire. So they basically have a very similar chemical profile with our tire leachate. So should we use them? Are they actually safe? This has actually been debating for a while, and until earlier this year or later last year, I think it's this year, there's almost like a smoking gun evidence from Europe. So there's a kind of like a soccer player in Norway. One of them are tested positive for a stimulant. Actually, the stimulant is in this picture. It looks like this one, right? It's a monoamine. And they're like, oh, we didn't use this thing, and the concentration is not enough to give you like any performance enhancement. Why? And they realize, oh, like they read this paper actually from us in 2023, and they found, oh, this is actually from the rubber because in the winter of Norway, they play indoors. And this indoor field is with all this like chrome rubber. So this is the good evidence that it actually get into human and it can be detected. So this is still kind of like something and ongoing because you right? Like if you later previously you are saying, okay, I don't care about that, but what about you get tested positive for a stimulant because even you don't use it, right? So that's some of the ongoing research I have in my lab as well. This is more like NSF supported research, so we are not that directly into human exposure, but we are trying to see how the chemistry of rubber change when they are on the playground. So you can see basically this is the ratio of the parent compound versus the transformation product, 6-PBD and 6-PBDQ. Over this time course, right, these are the samples we collect from different age of field, right? You can see they have a pretty big difference, right, around like 1 year to 3, around like 2 year time period. So this is telling us, right, like even when the rubbers are on the field, they are still going through some pretty active transformation. And this is not only right on the chemistry side. There's another lab, I believe it's Wayne State University. The toxicologist also tested the kind of biological response from the chrome rubber. They're also seeing the older ones and newer ones are different. Some of the toxicity are actually stronger in the older ones. So the right like for this project our kind of interest is like okay um what can we see about the aging process of the rubber when they are put in the playground, right? Can we see some of the markers? Because a lot of the field, right? Even you know where this is built, you don't know actually how old this is rubber. So it's hard to assess are they active or inert? Or to be honest, is there actually an inert phase for these things? So this is kind of like a lot of interest we have from the beginning. And we started to do that because both for our study and for this one, right, we are actually collecting samples that's already been aged on the field. We are now seeing the same rubber from the beginning. So we started to do our own controlled experiment. So this is actually done at a rooftop of a Northeastern University building. And we are doing both like outdoor, which is less controlled, and also like an indoor artificial aging with, light and all this like kind of like simulation. So we have dry, we have wet, and we also have different rubber samples because, right, like their kind of age are different, so their chemistry are very different. So two of them are kind of like a tire, right? So this is almost like assuming the tire was not too old when you put them on the field. So this is a cryo-milled tire tread as well as a tire wear particle. So these are the newer ones. And also we have a 0-day chrome rubber sample that we collected from a field when it's being installed. This is from New Hampshire. So these three samples will give us different kind of pictures of how this process is going. And for the outdoor aging, because we have limited time to using the roof, so this is like about 3 months. But then the indoor, right, this is kind of like matching the time frame, but we can intensify the light, so making this a little bit faster. So that's like 18 days. And this is a setup. So it's pretty simple, right? You need to have a coarse plate, and then you can put the rubber in there. And when it rains, we actually have this funnel to collect the rainwater and pour in there. So this is simulating the rainy like condition. And for our analysis, this is the workflow we use. So from the samples, we go to both quantitative analysis. So this is not non-target. And then the bottom one is non-target. So basically, for the non-target, this is more interesting because we're not only just trying to see what is what, but we prioritize things based on their time trend. So if there's something going up, something increasing in the process, we're able to grab them out. And also we use a method called molecular networking. Later you'll see for the drug, we use the same technique. This will actually help us to find analogs from known chemicals. And finally, for something that's really unknown, what we did is in silico fragmentation. So this is almost like a de novo identification for molecules. So this is kind of like the result, right? Like if we are only using suspect screening, we are seeing about 5% of the features. So it's not a very good identification rate. But then we use the time-based cluster to see, okay, can we narrow down the scope for interest chemicals and we can go hard on them, right? So this is the cluster we try to classify. So basically we have four types. The increasing one, which are the first, right, like one on the top left, and also some of the intermediate, because you will see chemicals going up and down. So they are being formed and then degraded. And then also we have the degrading ones like this, as well as the ones without a clear trend, like the last one. So if we have something that's like continuously going up, or we have something going up and down, these we think are the transformation product. So we actually have a lot of them, like more than 500 from the new tire like powder, the CMTT. And then among these 500, right, like 190 are increasing. So this is still pretty big, but because we have the three different rubber samples, we can actually overlap them to see, okay, no matter the age of the rubber, if we see this thing going up, right? So by doing that, we are actually narrow down this to 37. So we say, okay, these are the aged rubber markers. No matter when you put this on the field, if you see this one kind of like showing up, this is a sign of aging, right? So that's one of the kind of a pathway. And then from that narrowed route, we are actually also doing the GMPS, the networking, to see if we can increase our pool of identification. So you can see the pink ones. This is the thing we already identified with pretty good confidence. Then we can expand to this orange ones. So these are the potential transformation product. Some of them we have a relatively good structure information, but some of the others we don't. So you can see this is the Marcouche structure. So basically means we know what the functional group is, but we don't know where it's located. Things like those. So with this method, we are able to increase the identification rate from 5% to 11%. Still a little bit low, but we doubled it, right? So this is giving us more knowledge. What are the reactions happening when the rubber is aging? And some of the more interesting findings coming from this cluster, right? So we found, okay, there's a big cluster in the network that is like showing all these potential TP, right? This like orange, but no pink ones. So there's no identified chemicals in there. What are they? And turn out to be this is a group of things related to anti-ozonant called um, TMQ. And the reason we don't have identified things is because a lot of these are oligomers. So this is something we thought, okay, when the industry is putting this into the rubber, they're using just single small molecules. But what actually they putting into there is a cluster of multiple of this, right? So you have this dimer in there. This is like almost like an impurity, but actually these things could potentially be the majority of mass they put in there. So we have a lot of dimer, trimer of TMQ. That's why we have this great cluster. And even more interesting, some of these dimer and trimer, right now we know they're TMQ related. We can put them into this cluster. Some of these things are actually solved, right? Like a puzzle from a previous paper. So this paper, I believe, is from Sweden. They are kind of like seeing, okay, what chemicals, if they are present in water, can go from water to fish, right? Lumpfish. What chemicals can bioaccumulate? So you can see all these things, they know what they are, but then this later two, these things with two nitrogen, they don't know what they are, but these are actually in the blood of the fish. These are bioavailable. And they thought this is something about, right, like similar to PPD because you have two nitrogen. But we realized, oh, these are actually not PPD. These are the dimers from TMQ. We checked the spectrum from their paper and our paper actually matched pretty well. So solve the puzzle, right? And that is telling us, okay, these group of things, we have a very little knowledge, but they can go into biological tissues easily. Okay, another one from here is we also find something, right? This is in the 37 aging marker. You can see this line of this chemical is going up. So it means when the rubber is aging on the field, this thing is increasing. And it's a pretty big molecule. It's 544. And we also find that the spectrum of this guy is actually a quite interesting combination of a bendoyl derivative and our TMQ dimer. So you know the part of this molecule, but you don't know how these things are melt together. But the important thing here is, right, when it's on the field, we are seeing smaller pieces getting together to becoming bigger pieces instead of only degrading. So this is kind of like a little bit alarming, right? Because it's telling you the rubber, when they're on the field, are still quite active chemical reactions going on. Okay. So that's enough of the rubber story. Okay. So the other one is where I would say, like in the later slide, you will see this is where lead us to the drug story. This is the water contaminants. So the major topic here we have is a group of chemicals called PMOC, persistent and mobile organic contaminants. So you can see some of these examples are, right, metformin, or if you know some of the short chain PFAS, these are actually the PMOC. So these are the chemicals very hydrophilic. They don't go to the lipid very easily, but they are very soluble. And a lot of them are transformation products. So this is the work done by Kat. So you'll see later the drug project is also her work. The reason we are interested in PMOC is we realize this is the group of chemical that our current water treatment system don't do very well at removing. So some of these actually go into our drinking water and the natural surface water. Right, like some of these are actually coming from our disinfection process, right? Things called DBPs are actually quite polar. So they all fall into this group. And the trait of these are, right, like they're very mobile, right? If you think about what our water treatment plant did, a lot of them are sorption as well as the bacteria doing this degradation. But if your molecules are very hydrophilic and also having some functional groups that's not very tasty for bacteria, they just penetrate through. So these are the groups. And these are potentially risky to human health because, right, if you think about the drinking water, right, like even at a lower concentration, we are exposed to these every day into our internal system. So that's why we are paying more and more attention to these, especially in the Europe, they started to doing this, but in the US, this is still something pretty new. And you can see this is one of the reasons previously why people haven't been able to analyze because the more conventional method like GCMS or LCMS, they are actually looking at more like hydrophobic or in the middle kind of contaminants. But for the PMOC, we really are looking at these like log KOW, which is an indicator for hydrophobicity, like very low or even negative range. So there's an analytical gap, right? You simply don't see them, then you don't know how they behave. So the sample prep and the chromatography would be quite important if you want to see these things. And also, right, like there's a lot of other things when you are able to analyze them, right, like their transport, their fate, their toxicity. A lot of these are kind of like still away from our current body of knowledge. So what we did first is develop a method. So we tried different chromatography. We tried, right, like use the standard on the right. So these are kind of like our model compounds. It's not necessarily saying these are the things will be detected most frequently, but we just know these things and they're where you can see their property are quite in the range of PMOC. So we use these things to develop our method, and we can do both quantification and non-target analysis based on our high resolution mass spec. So here's some of the developing process. I will not be, going to very detailed. So basically, you can see, right, like we have been able to detect some of these things in our drinking water, but generally, our drinking water is still pretty good, right? Some of them are right? The TW means tap water, BW means bottled water. So you see a lot of ND, which means not detected, smaller than LOQ, which means it's you detect, but you cannot quantify. So generally, we are good. Some of the water sample, for example, there's one where like a well water actually showing some of the artificial sweetener, which is not good. But other than those, right, we don't see a lot of these. And TFA, which is a short chain PFAS, this is kind of like getting everywhere, but this is a known problem, right? Like we don't remove these things very well. TFMS instead don't we don't see like a high level, so this is actually good. We also did some of the analysis on surface water. This is where we see a little bit more detection because surface water is naturally not that clean, right? So we kind of getting a right three batch of samples just from the Charles River, right? Easy for Northeastern University in dry days. And then we kind of like quantify these analyze. So these are the things we saw, right? So basically like some of those are pretty expected, right? Caffeine, that's from coffee or chocolate or other things, right? And metformin, which is the diabetes drug, we also see it quite abundant in some of the samples. So generally, right, it shows us, okay, the method is working and we can see some of these things in water. And if we compare, right, drinking water and the surface water, so the PFAS levels are quite comparable. So I guess it shows like they are just like very prevalent. Like removing them is still kind of like challenging, but it's not the focus here. Okay, so right, this is kind of like prioritizing things for drinking water, right? If the water authority want to know what's the most important things, so these are what we give them, right? A lot of right, like potentially disinfection byproducts, some PFAS, and also some artificial sweetener. So these are the identification. So we also have some very new identification over here. We don't know where they're coming from because there's no data. We also try to prioritize this based on the knowledge we have, which is the the x-axis, as well as the detection frequency, which is the y-axis. So you can see a lot of things, right? If we have something that's like frequently detected, but we don't have a lot of knowledge, then this block is the most interesting things. So you can see a lot of things with a lot of amine groups or amine group or sulfate group are over here. So these seem to be the most common functional group that gives this polarity, this like mobility to the chemicals. So for the future work, we will pay more attention into things like these. Okay, so probably linking to the drug side, right? So this is where we see some of the more interesting things in the Charles River. So this is a drug phase 2 metabolite. But if you see something like this, it's not concerning, right? Because this is just in OTC, right, drug. And some other things, I think this is also like a commonly used drug, like a metabolite. It's not too big an issue. But, well, these things, some of the, right, I think the venlafaxine is like a more prescription drug, right? This is where you see some of the more interesting things. And then the most interesting sample, this is actually coming from the Alewife Brook wetland. If it's in the brook, if there's a known CSO problem, combined sewer overflow, so I will not be surprised. But the wetland should be actually a isolated stormwater treatment system. But in here, we see a lot of cocaine. So you can see this is the mirror plot again, cocaine and the cocaine metabolite, right? And if you look at the peak area, this is not quantitative, but kind of like giving you an idea, right? We spike our internal standard at about like 200 nanogram per liter. But now we're seeing the cocaine and the metabolite as a similar response. So that's potentially pretty high. So, right, because we keep seeing this, right? Like I was like, okay, we should probably do some drug analysis with expert in this field. Our right skills can actually help. So this is again Kat's work, right? Like we have the sample from Tracy and Abby last year. We are trying to see, okay, what we can see from these. So we have 10 samples. We actually started with aqueous, right? Those are the easier ones for us to analyze. We did a dilution because, right, the samples can be a little bit intense. Then we use the same analytical flow for non-target. And so you can see, right, like this is kind of like a preliminary work. You will see all these quantitative information are based on peak area because we did not use a calibration curve to quantify them. So big picture ideas, right? So the first thing is we are seeing this like diversity of chemicals we can see from the drug samples. We are seeing more than 400 from this 10 samples. These are after the blank subtraction, after the replication filters, and peak quality filters. So these are the pretty good chemical features. So you can see, right? This is the heat map. We can see how they look like. Some of them are pretty intense, some of them are not. And then you can also see we can tell them apart by their different like kind of like features. Some of them are going pretty far away because they have higher complexity or they have some specific like, plastic related chemicals. So this is telling us, right, if we are trying to track the sources, we can actually use non-target to do that, right? Like both of these cluster method and PCA can help us do that. Another big picture idea is we also apply the molecular networking to these samples. We want to see, okay, if there are something that's like a known drug of abuse, then can we see the analogs of it? So this is quite interesting because, okay, we see heroin cluster, right? This is heroin cluster. So a few samples, but in general, right, if you see the nodes, right, each node representing one compound, right? Like heroin is over here in the middle, morphine is over here. This one not like too many. But then on the right, this is the fentanyl cluster. So you see a bigger diversity of nodes, more nodes, as well as you see the different color. Later, you'll see the detection frequency of fentanyl is pretty high. So almost in all these samples, you see a little bit of fentanyl. Um, okay. So after the big pictures, let's go through some of the interesting IDs one by one. So fentanyl is this guy, right? And also for ANPP, if you look at the structure, it's basically like a backbone of fentanyl, getting rid of this hydrophobic cap. So both of them are detected in like almost all the samples, and you can see that these two have a pretty good pattern correlated, right? So basically, whenever you have a fentanyl, you have for an AMPP because it's a precursor for fentanyl, right? So if we are not detecting this one, we can also use this as a tracker. Another group, right? These are something when we discuss with Tracy, we realize, okay, you already know these things. The fluorinated analogs of fentanyl, they are also discovered in the samples, but they are not as frequently detected, right? They are not in all. They are only in 4 and 3 out of the 10 samples. And then there's a great example of true unknowns, right? Like the previous ones are, we have some suspect or we have a spectral database, but these true unknowns, right? Like for example, this guy, there's no database match, but we see it in a few samples, and it turned out to be I believe it's a byproduct, right? You can see our computational software give us the structure. Two computational. The first one is our in-house, the vendor software. The one below is a open access software. Both of them giving the structure, so it's pretty solid, like a identification. If you look at the structure, it's not really aligned in the same way. So I believe it's kind of like a byproduct because you are putting the different part in a different order to get that. Is active or not, I don't know, but it's going to be another indicator for this synthetic process. Some other things, right? This are the classics, right? We do see methamphetamine and heroin in some of the samples, but not really that many. So meth are in 3 samples. In heroin, I believe it's really just abundant in one of the samples. So potentially they are being replaced by fentanyl. Potentially, I don't know. That experts here would know better. So another interesting one, right? Hopefully this is not too cringy. I was watching The Pit as well, and then when this scenes show up, right? They are going to the park for a girl was using xylazine, and I told my wife, "Oh, we actually saw that in the samples that Tracy gave us." And xylazine was in, I believe, 8 of the 10 samples, and then another veterinary drug in 6 of the 10 samples. We can see them. but I don't see like a very kind of like a good pattern. But that's indeed kind of getting a little bit more frequent these days. And some other things that's also learned new for me because we see lidocaines and procaines, I believe, in the samples. This is for, right, if they're injection, this is like a analgesic, right? So new knowledge for me. Okay, so that's most of the interesting stuff, right, data we have from our work. Some of the things I want to kind of like because I'm following the drug of abuse work a little bit. So these are some interesting work recently. This is from a friend. Basically, what they did is very similar with what I wanted, but they already published this. This is from Fudan University in Shanghai, and they are developing a workflow called fentanyl hunter. So basically, they use some of the molecular network and also the experimental to kind of like getting them a very big database of potential fentanyl metabolite and the fentanyl byproduct and all these analogs. So they put this into a right like a use of like screening work. So you can see again, right, this is the molecular network they used in this process. And when they have this database, they apply this not only to their local water sample, right, but also to the open access data all over the world. So you can see this map. They see where the fentanyl analogs and fentanyl metabolites are found all over the world. So this is a very cool work in Science Advance last September. And then another two actually from this year, I've seen this study in ACS where I believe it's Louisiana State University researchers, they found nitazene going pretty high after some special events like Super Bowl or Mardi Gras. They can see the spike in wastewater samples. So nitazene might be another hot batch of chemicals that we should be paying attention to, I guess. But the concentration they see for now is still not that high. And another interesting work is computational. They see why fentanyl is so potent and right, like hard to reverse. It's because fentanyl can go through the membranes much faster, 100 times faster than morphine. So I thought, heroin is based on same mechanism. I don't know, but yeah, that seems to be an interesting read. Okay, so these are my slides. These are my team. So Kat, this is Philip and Madison. So Madison graduated. Kat did most of the work on the water, and Madison did the rubber stuff. So and the undergrads over here, and we are collaborating with Tracy's group for sure, and some of the previous collaboration with UW. And the funding sources are from the Northeastern startup and the NSF funding for the rubber stuff. Thank you.
That was wonderful. Thank you so much. I'm just coming over here for to chat a little bit more. We would, I think we have time for some questions and conversation.
Hi, this is Ju Park. Can you hear me?
Yep.
Oh,
Yeah.
awesome. So nice to meet you, and thank you so much. This was the most exciting chemistry talk that I've ever been to.
Thank you. Flattered. Thank you.
I work with Tracy and other people at the COBRE, and I think that there are so many parallels between environmental chemistry and some of the work that we're trying to do with drug checking and drug surveillance.
Cool.
My quick question, since we're running out of time, if there was no restriction on funding or, time, what would be the fastest turnaround time for testing one sample, in terms of both identification of the chemicals through non-targeted analysis and then quantification? So that's my first question. And then the second question is, do you think that the role of AI or technology developments might speed up that process?
Great question. So the first one is, I think, right, like a fast turnaround, how fast we can do, right? I would say if the instrument, I think, yeah, imagine we have good funding, we have good instrument, right? Like instrument don't breaking up, that's very important for us. So I think in probably a few days to a week, we can get good, right? Like because you need to have the sequence run and the data analysis will either take me or my student a couple of days, if not like a week, to go through. So that's gonna be, I think, the turnaround. It's not gonna be super, right, like slow because the extraction, the instrument analysis generally will only take up like 2 or 3 days, right? And then the rest in the week would be data interpretation.
Thank you so much.
The second question is,
Yeah.
oh, AI. Yes, that's definitely helping us a lot. I was testing some of the identification myself, and also, right, like if you kind of like having some of the spectrum, you tell how, right, like the nitrogen rules, the kind of like molecular like chemistry rules work. That is definitely helping. And also we have this like different like de novo workflow that's developed in MIT. I have a collaborating lab computational chemists in MIT. So they are really doing cool work, and we are going to see how that going to help. But it's definitely like the AI is helping on the non-target identification side, because currently you're seeing, okay, our identification rate is still 10%, the best sometimes like 30%, but hopefully AI will help us go above 50%, I would say.
Awesome. Thank you so much.
Thank you.
Yeah, go ahead.
I guess start with, so you looked at factors involved with aging of the samples of the rubber samples that were correlated with the toxicities you saw. Yeah, versus different receptors. Did you see any compounds or identify any that were inversely correlated with that toxicity, things that were seen to be protective or prevent degradation or prevent the emergence of the causative agents?
Yeah, that's a great question. Because for this aging part, we only see the trend of the chemistry side, but we haven't been able to link that with toxicity. So if we do, I think there would be a bunch of them, I would say. But also, I think the caveat is, the Wayne State University group, they actually use multiple, I believe they use 4 different assays. So for each of them, you will have a specific group, right? For endocrine disrupting, for thyroid receptor activation, the group would be different. Yeah.
And what conditions had you already tried? You had previously established or shown, at least for 6-PPD, that it seemed to be a ozonolysis or some sort of ozone-related chemistry, radical chemistry. There are, of course, some rubber and plastic additives that have antioxidant properties, as you showed, UV protectants as well. Were they generally, I guess those would be the ones I would think be perhaps protective.
Yeah.
Do you think those, when you're trying to age these samples, are you trying to produce those radical oxidized products as well then?
Yeah, so I believe for the past, right, experiment, we didn't try to control where they go. We're just say like, okay, if things are put on the ground, what would be? So the question is, right, like, because for 6BD, right, some of these are like antioxidant, like what kind of like direction we're trying to, right, like push this through to make them either less or more toxic, right? But I believe we are able to see some like common reaction patterns. Like for example, oxidation is definitely something, and then hydroxylation, if you see the wet samples, are definitely something that we see a lot of commonality through those. But I think the unexpected one were the melting, because we are seeing, okay, the average molecular weight were not degrading as we expected. So that's actually something new, I would say. Yeah. But we are not controlling the direction of the reaction.
I was curious how where the if you feel like the conversations at the policy level, you've seen a lot of seems like a lot of discussion around the environmental contaminants and potentially changes that could happen at the state. Do you have a sense if there are things that we could do at the state level on some of the drug related risks that we might see on making the supply safer or not, or raising awareness around? I don't know if you've seen wastewater be for instance, a surveillance technology that people are talking about, or
yeah, totally. I think, Ray, I haven't have a lot of experience on the policy level, but my naive thought would be if we have some of the early warning system, either is on wastewater, right? Actually, the wastewater analysis during COVID, I believe we have a company called BioBot in Boston was actually doing pretty good work analyzing the a virus, right, DNA. So similarly, this can be applied to the small molecule chemicals like drugs. So I think some kind of like a early warning network like that would be very helpful, right? If we are seeing something new is getting kind of like emerging, right? Or we see some of the things we know are kind of getting up, there should be something, some action to be done. And I believe, right, I don't know how this can be work like logistically, but if the street drug kind of checking can be also right another kind of an entrance for this warning system, alarming system, I would say.
From your lips to the policymaker, endpoint. That would be awesome. I love it.
Thank you. Yeah, but definitely like we are not the expert in the public policy side, so we definitely need to work together. Yeah.
Well, it seems like we're really at the edge of a lot of opportunity between what we think we can put together and understand from wastewater, from stormwater, from wetlands, and sort of thinking about the impact of substances in humans as well as in animals and fish, and future generations too. But maybe monitoring many different things, the water, but also maybe the drug supply is part of our environment.
Totally.
That's great.
Great.
I will close this up just because we're at time, and I know we're very grateful that everybody joined us. Thank you so much for joining us.
Thanks.
And we'll see you next time. Thanks.